overview of this paper that came out recently. That was all of my PhD and thesis work with Hayden Kissick at Emory. And then now I'm a postdoc at MSK with Andreas Scheitinger, but I'm very happy to talk about this work. So hopefully, if you're here, you're interested on CD8 T cell exhaustion and CD8 T cells in chronic antigen settings, such as cancer and chronic infections. And so for many decades now, we've known that CD8 T cells gradually lose the ability to proliferate and to produce cytokines. And this is an older from John Ware's group, but you can see that as CD8 T cells are exposed to chronic antigen, they lose the ability to proliferate as well as to produce many cytokines. And that coincides with increased expression of inhibitory receptors. But then more recently, obviously now with immunotherapies and other things that we know for anti-PD-1 and anti-CTLA-4, we know that some of this function can be restored. So CD8 T cell exhaustion is not, as permanent as we thought. But even more recently now, and this is what our lab really focused on, is this new model of CD8 T cell exhaustion where you have stem-like and terminally differentiated, more effector-like CD8 T cells. And this was originally published by Rafi Ahmed's group, as well as many others. It's now been shown in mouse models, in human disease, cancer, and chronic infections. So in this model, you have this stem-like CD8 T cell that is activated. It can self-renew. It is proliferative. It is denoted by TCF1, which is a transcription factor. These cells are PD-1 positive, but they're extremely functional. They can proliferate, they respond to PD-1, and so on. And why these cells are so important is that they can differentiate into these more effector and more terminally differentiated cells. So these are now TCF1 negative, which is one of the main transcription factors we use to study these cells. They express very high levels of PD-1 and now have upregulated the expression of other inhibitory receptors such as TIM3 and CTLA-4. And more importantly, these cells now express granzyme B, and they're the ones that are really the effectors. So in this model, you really need both subsets, the stem-like CD8 T cell to be constantly producing these more effector cells. And so this has now been shown to be a model that exists in human cancer, but is also really crucial for the anti-tumor response. And this is just a small part of the work that has now been done. But these stem-like CD8 T cells predict overall CD8 T cell infiltration in tumors, as well as the response to patient with immunotherapy. And our group has worked on these now, as well as this slightly newer paper with our collaborators in Rafi Ahmed's group to show that there's antigen-specific stem-like CD8 T cells in HPV-positive head and neck cancer. So in our lab, we're extremely lucky that we get to study these human kidney cancer, and we get many patient samples. So when we looked in a human kidney cancer tumor and we gated on CD8 T cells that are activated based on PD-1 and CD45 RA negative, we find the same two subsets that I just described. So we have this nice stem-like subset that is TCF1 positive, TIM3 negative, and then this more effector terminally differentiated subset that is now TIM3 positive and TCF1 negative. And you can see that the TIM3 positives are very high for granzyme B, and they're the ones that we really think are the effectors. And then also we use this mouse model that I will be talking about a lot in this talk, which is a TRAMP-C1. It's a syngeneic BLAK6 prostate cancer cell line that we engineered to express the LCMV glycoprotein so we could use it as a model antigen. And in this tumor model, we can look at the tumor-specific GP33 positive CD8 T cells that are PD-1 and 44, so they're activated, they're tumor-specific for the LCMV GP, and we find the same two subsets. So we find the stem-like CD8 T cell that's TCF1 positive, as well as this TIM3 positive cell that expresses slightly higher amounts of granzyme. So we do think that these subsets are really important in cancer, and we have all different kind of models to study them. So when I started, we knew that these subsets exist within the tumor, but we didn't really know what is the origin of these stem-like cells that we think were so important for the response within the tumor. So we had several hypotheses. The first one is that these stem-like CD8 T cells are coming from the tumor-draining lymph node. They might be activated there. They're migrating into the tumor and then differentiating. A second hypothesis was that both of these cells are activated and differentiate within the draining lymph node, and then they both migrate into tumor. And then lastly, these cells could just be activated within the tumor directly and then just establish the response that way. So my whole PhD was trying to figure this out basically, and so this is where this paper really came from. So we first wanted to look in our human samples. So we looked at prostate and kidney cancer tumors, as well as the non-metastatic tumor draining lymph nodes. So once again, gating on CD8 T cells that are activated based on PD1 positive and CD45RA negative. And we can look at the phenotype of these cells. And so within the tumor, as I've previously shown, you have this TCF1 positive stem-like cell and this TIM3 positive cell. But very interestingly, within the lymph node, you find that the activated cells, the vast majority maintain TCF1, and you have no TIM3 positive cells in most of these lymph nodes. So we really have looked at many prostate as well as kidney cancer, non-metastatic tumor draining lymph nodes, and the vast majority of these cells are in this TCF1 positive stem-like phenotype. So I'll call these cells lymph node stems from now on, just for clarity. And so we've characterized these cells from the human tumor draining lymph nodes extensively, and there's a lot more in the paper, both RNA-seq and DNA methylation, but it's all very similar story. These cells do not express markers associated with effector differentiation, such as CD39, which you can see is extremely highly expressed in this terminally differentiated TIM3 positive cell, or granzyme B. So they really are in this lymph node stem kind of phenotype. So our next question really was, are these cells that are activated in the lymph nodes clonally related to the populations within the tumor? So to do this, we sorted and did TCR sequencing on patients where we had matching tumor draining lymph nodes as well as tumor. And so this we did in 5 kidney cancer patients as well as 4 prostate. And you can see that in every patient in the green is TCR overlap between the lymph node and stem-like cells within the tumor. And there's quite a bit of overlap in every single patient that we've done, which really shows that there is a clonal relationship between the lymph node and the stem-like population within the tumor. And then when we analyze this further, we find a lineage between all 3 subsets. So here on the x-axis is the TCR frequencies in lymph nodes, on the y is the stem-like from the tumor, and then all of the greens here show TCR overlap, and then the red filled in circles are also the ones that overlap with the terminally differentiated subset. And this is just quantified in every patient on the right. But you can really see that there, in fact, is a TCR and clonal relationship between the activated cells and the lymph node, the stem-like and terminally differentiated cells within the tumor. So that was very briefly talking about all the human work that we've done. But just from the human data, it seems like these cells in the tumor draining lymph node and these lymph node stem phenotype are the ones that are clonally related to both subsets within the tumor. But the next question that we really wanted to get at is, how are tumor-specific CD8 T cells activated, and are they activated in the lymph node? So to do this, we went into mouse models. And so we used 3 different models. The first is an acute viral model of LCMV Armstrong, and this was really our positive control to see what an effector CD8 T cell activation and differentiation was. And then we used 2 different established tumor models. One is the TRAMP-C1 GP model, which is very slow growing prostate cancer, what I previously showed. And the other one was this B16 GP, which is a melanoma mouse model that expresses the same LCMV GP. And so for all of these models, we can transfer in the same T cells because we have P14s that are TCR transgenics specific for LCMV GP. So in all three models, we can transfer them in and look at the antigen-specific T cell responses. And I also labeled them with cell trace violet, which just tracks cell divisions. So we can look at extremely early CD8 T cell activation. So when you look after LCMV Armstrong infection in a lymph node, you see that these cells have undergone many rounds of division. They upregulate TIM3 very highly. They're also very high for CD44, PD1, which is upregulated upon T cell activation, granzyme B, everything that you could think of that's a typical CD8 T cell that's getting activated to a viral infection. And you can see they also downregulate TCF1 quite rapidly. But in stark contrast, in the tumor-draining lymph node, these cells are activated. They have undergone many rounds of division in both the B16GP lymph node as well as the TRAMC1GP lymph node. So you can see they've undergone many rounds of division. They upregulate CD44, they upregulate PD1, they're activated, but they fail to upregulate TIM3, they don't upregulate granzyme B, and instead they maintain TCF1 at this extremely high level. So they're really activating to acquire this more stem-like phenotype that is extremely different from what we think of as classical CD8 T cell activation in an acute viral infection, such as LCMV Armstrong. And we also have a lot more of the characterization of this very early activation, including RNA and epigenetics, also in my paper. But the main point was that these cells activate within the tumor-draining lymph node to acquire this more stem-like phenotype. They don't gain any type of effector phenotype. So we next want to see if these cells that are activated within the lymph node are the ones that are establishing the response within the tumor. So we did basically a kinetic study where we transferred in P14s into an established tumor and then waited for different time points afterwards. So you can see at day 3, which is the same time point I just showed previously, everything in the lymph node is TCF1 positive, TIM3 negative. None of the cells are directly activated in the tumor in this model, both in TRAMC1 as well as in B16. All the cells first go to the lymph nodes in these models that are subcutaneous. But then when you start looking later time points, days 4 to 5, you start to see these cells within the tumor. And so you start to see these transferred cells in a TCF1 positive phenotype that have not yet differentiated, but they have now made it to the tumor. And then when you look at later time points at day 10 and even later, you see that now you have this nice TIM3 positive population in both models, and these cells really have differentiated into all 3 subsets. But very importantly, at all of these time points, the cells within the lymph nodes have maintained their phenotype. They are still lymph node stem, they're still TCF1 positive, they have not differentiated, even if they've been in this lymph node for 2 weeks. So we really wanted to show that these cells are in fact the stem-like cell that is giving rise to all of these populations within the tumor. So to do this, we did a quite elaborate retransfer experiment where we basically made lymph node stem P14s by doing what I just showed you, transferring in P14s, waiting for 2 weeks when all the subsets are established. Then we sacrificed these mice, sorted only from the lymph node, CD44 positive, PD1 positive activated cells, retransferred them into a new tumor-bearing host, so they're constantly still seeing antigen, and then just gave intratumoral CpG because this is such small cell numbers to transfer, just gives them a little bit of a kick to migrate into the tumor, a little bit better to detect them. So to our great happiness, when I did this experiment, we could find these P14s within both tumor-draining lymph nodes as well as in the tumor. And we repeated this multiple times, and it's quite consistent. And when we look at the phenotype of these cells, we see really importantly that in the tumor-draining lymph node, they self-renew and they give rise to more of themselves. So you see this nice population of lymph node stems. And then once they migrate into the tumor, you see that they do in fact make up both a stem-like and a terminally differentiated subset. So these cells really are the precursors to the ones that we find in the tumor. So hopefully I've shown this quite briefly, but that in fact there are lymph node stem CD8 T cells that migrate into the tumor and then establish this response as a stem-like cell, and then they can differentiate to upregulate TIM3 and granzyme B. But throughout this work, we found it really interesting that this differentiation is only occurring within the tumor. And so in the lymph node, we mostly just find these TCF1 positive cells, but within the tumor, you have this effector differentiation. So the next question that we really had is what signals are promoting this CD8 T cell differentiation within the tumor specifically. And just for the sake of time, I skipped a little bits of how we got here, but we knew from other work and from other work from our lab that co-stimulation as well as TCR signaling are extremely critical for CD8 T cells for activation and differentiation. So we want to see if these signals were also necessary for these stem-like CD8 T cells within the tumor. And we went into this ex vivo model where we can CTV label kidney tumors and sort out the stem-like CD8 T cells based on PD1, CD28, and then 39 negative, and then culture them ex vivo in different conditions with low levels of IL-2. So we used CD3 beads, which would provide TCR alone, CD3 and CD28, which now gives some co-stim through CD28, and then CD3, CD28, and CD2, which is additional co-stimulation through CD2. And so super interestingly, when you take these already activated stem-like CD8 T cells from the tumor and you give them TCR signaling alone, they do not proliferate and they do not differentiate. You can see here, they're not diluting CTV, they're not upregulating granzyme B or CD25, same with 39 and TIM3. But once you give them some co-stimulation through CD28, you start to see these really nice cell divisions. They upregulate granzyme B, they upregulate 25 and CD39, and they're really starting to differentiate. And when you give them even more co-stim through CD2, you see more of these cells are now dividing and differentiating. And this is some just summaries on the right, but you really find that these cells that have already been previously activated, they're in the tumor, really need both TCR and co-stimulation in order to differentiate further. So one of the next questions that we really had is, where is this co-stimulation coming from? And could the co-stim predict CD8 T cell infiltration? So we went into our many human kidney cancer tumors that we've run over the years and looked at that. So patients with high CD8 T cell infiltration also have very nice differentiation, meaning they have a good population of these CD39 positive effector cells. And then patients with low CD8 T cell infiltration also do not have a lot of these 39 or TIM3 positive cells because this differentiation isn't happening as actively. So if we look at the major APC subset that is in kidney tumors, which are monocyte DCs based on CD11B, and look at the expression of co-stimulation, CD86 and CD40, we find that in fact, patients with high CD8 T cell infiltration in their tumors also had significantly higher levels of co-stimulation on their monocyte DCs. So you can see here, CD86 as well as CD40 is significantly higher. We also looked at other APC subsets. Here, CDC2s, which are gated on CD1C positive. And very interestingly, these co-stim is not different. CD86 is not different between high and low infiltrated tumors, but CD40 is in fact still upregulated. So this could, future work on CD4s or other mechanisms. And then also importantly, CDC1s did not show any significant differences in co-stim between high and low infiltrated CD8 T cell patients. So this seems to be other APC specific. So we also want to show that this is not only a correlation, but these cells are in fact functional. So we did another ex vivo experiment where we have the CTV sorted stem-like CD8 T cells, And we could sort different APC populations from the same piece of tumor. So it's all autologous. So we sorted monocyte DCs as well as CDC2s for some experiments, irradiated them, and then co-cultured these two cells only with low levels of IL-2 for 7 days. And as you can see, monocyte DCs, in fact, can induce the stem-like CD8 T cell differentiation. And you see that there are cells that are dividing as well as upregulating granzyme B and CD39. And that's pretty consistent in the few experiments that we've done. But it does show that these monocyte DCs and potential differences in co-stimulation on them are, in fact, crucial in vivo. And so finally, we did want to show that this is important in vivo. So we went into mice and back into our TRAMP-C1 GP model and transferred in P14s, waited for 5 days. So that's a time point where the cells are activated and they're starting to migrate into the tumor. And here we could give them CpG intratumorally, so activating all the APCs in the tumor and look at how the CD8 T cells responded. And so when you give CpG intratumorally, the lymph nodes don't change significantly, but within the tumor, you see this really nice upregulation of TIM3 and increase in differentiation of these cells. And then to show if co-stim was one of the driving factors for this, we gave CpG while blocking 80 and 86. And then we see that this really nice decrease of the differentiation with summaries shown here. So it does seem that activating APCs within the tumor can increase CD8 T cell differentiation, and then at least partially responsible for this is the co-stimulation through 80 and 86. So hopefully I've shown this rather briefly, but hopefully it made sense, that CD8 T cell activation in tumors and cancer really has two distinct phases. The first phase is in the tumor-draining lymph node, where you have tumor-specific CD8 T cells that are activated. They acquire this lymph node stem phenotype where they're PD-1 positive, TCF1 positive. They increase migration receptors, activation receptors. They're still very proliferative, but they're not very effectory or cytotoxic. They're really a lymph node stem. The second phase occurs when these cells migrate into the tumor, and we think from co-stimulation and TCR provided by dendritic cells in the tumor APCs, now these cells can differentiate into this more effector TIM3 positive cell. And so this is where you really get this granzyme B and effector cytokines, and these are the cells that you want in order to kill all the tumor cells. So we really think that these are the two phases that are occurring for CD8 T cell activation in cancer. And feel free to read the rest of the paper, which has a little bit more detail on all of this. But I just want to thank my PhD lab, who are absolutely incredible, and I miss them every single day. So watch out for what they do next. They're amazing. Our very close collaborators, Rafi Ahmed's lab, who are amazing, and we very grateful to work with them, all of the surgeons and everyone at Emory that makes all of this possible. Thank you to you guys for having me. And then if you have any questions you're not able to ask today, just feel free to reach out to me on Twitter or by email or anywhere else. So thank you so much, guys.
Thank you so much, Natalia. That was a really great presentation and really a lot of work. So if anyone in the audience has any questions, please raise your hand or I will unmute yourself. In the meantime, I would ask you one question. So did you ever try to do in your in vitro experiments to add another cytokine, such as I'm thinking of TGF-beta, because I think TGF-beta is somehow regulating this phenotype of these T cells. So I was wondering if you ever add any other cytokines rather than IL-2 or TCR-CD28.
Yes, that's a really good question. So we did add cytokines. We went in the opposite direction. We were adding more inflammatory cytokines. So we did do IL-12, and IL-12 doesn't compensate basically. So if you do CD3 with IL-12, it doesn't do anything. It maybe has a little bit of effect with low levels of co-stim, but it doesn't seem like directly on the stem-like CD8 T cells, there's a very significant effect. And we also did type 1 interferons, which was a very interesting phenotype. It prevented a lot of proliferation, but the phenotype overall was the same. But we didn't do TGF beta. That would be really interesting to do as well. We were going into the inducing differentiation path. So TGF beta could be interesting to look at if it's preventing it even with co-stim. But yeah, maybe my lab mates should do it now.
Thank you, Nataliya. So I don't know if anyone in the audience has more questions. If not, you can feel free to contact Nataliya. This is a great work. And I think we could go with Margaret.